Hello everyone, this is Amya, working as an assistant professor, Department of Zoology, Government Pascal College, Jayanagar. In this studio, in this video, I'll be explaining the Herdmania larva, which is a larval tadpole larva of Herdmanians. See, as were discussing the general characters and of chordates, so I said the Herdmanias or the Eurochordates exhibit indirect type of development, that is, which involves the larval stage during their development. So, in case of Herdmania, it includes the tadpole larval stage during the course of its development. After the formation of the zygote and the development of the egg hatches into the larva. So this is how the larva looks like. When you look into the characteristic features of the structure of a larva, a fully grown tadpole larva is highly motile and does not take food from outside. That is, it is a non-feeding form. So in case of these Herdmania, the larvae are non-feeding forms. It has an elongated body structure. The body is more or less oval in outline. The body of the larva is quite oval in their shape. It is distinctively divisible into two regions, that is the head and the tail region. See, we can make out the head and the tail regions are there. The whole body of the larva is covered by means of tunic, as in case of the adult also. In case of the larvas, the whole body is covered by means of tunic. In case of the head is elliptical in shape and has three adhesive papillae. Here in the anterior end of the head region, we can see three adhesive papillae. So in that, the first one is mid-dorsally placed, where the other two are ventral laterally placed adhesive papillae are there. So these adhesive papillae later on during the course of metamorphosis that will be helpful to get attached to the substratum. The tail is laterally compressed in case of this Herdmanian larvae, and they are provided with the caudal fin. So you can see they are having the caudal fin, and the ventral and the dorsal fin are continuous with the caudal fin. So they don't have any demarcation or the separation between the ventral, dorsal, and the caudal fins. Caudal fin are there, and the ventral and the dorsal fin are continuous with the caudal fin. So these fins are supported by the stirae. So stirae is like that is like a precursor for a fin brace, which you'll see in case of fishes. The central nervous system, which is situated dorsal to the notochord. So dorsal to the notochord, we can see the central nervous system. It enlarges anteriorly into the sensory vesicle, which opens into the pharynx and the mouth by neuropores. So the central nervous system which opens to the mouth by the neuropore. A single median eye can be seen uh, and that median eye is containing the retina and the pigment layer. Cornea, lens are present in the median eye of this what is that, the tadpole larvas. So an autocyst, here we can see, wait, yeah, the autocyst, which is a sensory vesicle. The autocysts which are there in the sensory vesicle, autocyst, otherwise called the statocyst, are there. They act as a organ of balance. So these are usually situated in ventral side and are usually in a very rare, they bears ocellus. So usually they bear ocellus. There is a slender nerve card, is not much differentiated, but is hollow throughout. The nerve card is composed of single layer of epidermal cells. So the nerve card is there. The notochord is restricted only in its tail region. That's why these are Eurochordates. The notochord is restricted to the tail region. It extends anteriorly up to the pharynx region and is ensheathed by gelatinous material. We can see segmented muscle bands. So here with respect to the kind of lines are showing here. So those are the segmental muscles are present in the tail region. Here we can see the segmental muscle are there in the tail region. Well, the mouth is present and the alimentary canal is rudimentary. They have the mouth opening, but in these cases, the alimentary canal is rudimentary. Here, the pharynx is sac-like structure, which is well developed. It bears a fully developed endostyle and a pair of gill slits. Two pairs of gill slits are there. So here we can see a non-functional heart with epicardia, which lies beneath the endostyle. Yeah, here the heart, which is non-functional, lies beneath the endostyle. The paired atrial sacs are there. Just after hatching the tadpole larva, uh, atrial sacs, whatever the paired atrial sacs are there, that becomes positively to the phototactic and negatively to the geotactic. So these are the morphological structures that we do see in case of the Herdmanian tadpole larvas. So here they have given, or here it is tail, vertical section of the tail of tadpole. Here you can see the dorsal fin and the ventral fin. And within that, within the ectoderm, we can see the presence of the notochord, dorsal lateral muscle bands, and the lateral muscle bands, ventrolateral muscle bands. So the notochord can be seen in this larvas can be identified with the transverse section of this Herdmanian larva. So to summarize this session, so in this session, we discussed the morphological features of tadpole larva. As I said, the Herdmanian tadpole larva, which is a larval stages of Herdmania, which is there in the urochordates. They have the chordatic features like the notochord and the nerve card confined to the tail region. And we can see the free swimming larvae in case of the Herdmania. But how exactly during the metamorphosis, how this free swimming larva will get metamorphosized into a sessile Herdmania? Because in our last video, we discussed the adult Herdmania, which is sedentary in nature. So how come the free living larva will get metamorphosized into sedentary adult will be seen in the future videos. Yeah, these are the references that I referred for the particular video. Yeah, thank you.